Okay, so in the second half of this set of PowerPoints now, as I said at the conclusion of the last video, we're going to shift into looking at things a little more at the cellular level here. So the question there is, you know, we know what large cortical areas are involved in learning and memory, but what is actually happening in the brain that enables the brain to change, right? That's what we're suggesting, right? Is some form of neuroplasticity is involved when, if in order for the brain to store information that it did not previously have, there has to be some sort of change in the brain. In order for behavior to change, there has to be some change in the brain. So that means that we need to understand what's really happening here. And of course, as we learned from Lashley, it's not going to take the form of what we may have considered to be something like an engram, right? But let's come back and talk about Donald Hebb again. So if we remember from the chapter on neurodevelopment, we have Hebb's law, right? We already learned that that in childhood, where we receive a very rich, stimulating environment that actually activates synapses, that the repeated activation of those synapses keeps them alive. And if they are not used, then they will end up being pruned away, and or even sometimes neurons themselves will just kill themselves off through apoptosis. And so this principle that comes from Hebb that we call Hebb's law, shorthand neurons that fire together, wire together, is that if we repeatedly activate the synapse between neuron A and neuron B, that synapse gets stronger in some way over time because of some sort of metabolic change that occurs, right? So first question, of course, is what is that metabolic change? So we're going to go back to some very classic studies and looking at individual neurons here by looking at sea slugs. One of the reasons why these sea slugs have been studied is that their neurons are actually large enough that you can kind of almost see them with the naked eye. And so then you can kind of study what's going on here. So one of the things that we see we can do in this kind of research is to create a habituation response. So a neuron will respond to a stimulus, right? So but what you can have happen is that if you repeatedly apply a stimulus over and over and over again, especially if it's a reflex, right? So if it's like in the case of like imagine the knee-jerk reflex, and if you repeatedly tap the tendon over and over and over again, eventually we might see habituation so that the strength of the reflex decreases, right? So it just stops responding. Now, in the case of the sea slug studies, they're looking at a muscle contraction from a irritating stimulus applied to the foot of the sea slug that causes muscle contraction, right? But we see that if we repeatedly activate this reflex, again, we see that there is some form of habituation occur. But the question is, you know, what's actually happening at a neural level, right? So is it, for example, muscle fatigue, right? Is it something happening in the actual muscle fibers or myofibrils or sarcomeres, if you remember back to that chapter, that causes the muscles to, you know, maybe become depleted of calcium and therefore no longer able to really produce a strong muscle contraction? And the answer to that is no. Because if we directly stimulate the motor neuron, and then what we're going to find here is that you can get the muscle to contract normally. So we're not thinking that there's something happening in the muscle here. And what about the sensory neuron, right? Is it possible that the sensory neuron has become fatigued? But if we observe its actions, we find that the sensory neurons are in fact responding to the stimulus. So if it's not the sensory neuron, and if it's not the muscles, and if it's not the motor neurons, what is it? Maybe it's in the synapse, right? So it's not in a neuron, right? This change in behavior, this change in response, it's not happening inside a single cell. it's happening at the level of the synapse between two cells, right? There has been a change in this synapse where the sensory neuron maps onto the motor neuron. And so that's how we're going to explain habituation. There's also an opposite effect that can be created as well. So habituation is a decline of responding over time due to repetition. But sometimes you can create sensitization as well, so that you can make it generate a response that is becomes even stronger over time, right? As the response becomes sensitized, right? And as it turns out, there are other neurons called interneurons that help their synapses with the sensory neurons are increased now, and so there's this facilitating effect as a result of this as well. So clearly, the answer is when we think about engrams. Usually there was some thought about this memory being stored inside a neuron somewhere, but what these sea slugs studies are telling us is that at least when these fairly simple learning mechanisms of habituation and sensitization, the answer is to be found in the synapse, which of course echoes Hebb's law that suggests that it is something about the synapse that changes over time when you have repeated activation between two neurons. So let's look and see a little bit more about what specifically Hebb had hypothesized. So he says that the first thing that happens is that you're going to have an experience. Like, we can think about something like a classical conditioning experience where you're going to have a stimulus that is going to activate some neurons in the brain, right? And so the exposure to that stimulus creates what he's describing here as the short-term activation of this collection of neurons, right? So you get this little burst of activity, right? Now, because this is a pattern of activity across several neurons, again, it's not just about the neurons, but it's about, there's an important thing to talk about with the synapses between these neurons. So if we repeat this stimulus over and over again, then you're going to have the same activation of the same neurons again and again and again. And it becomes, therefore, a lot easier to recreate that pattern of activity across these neurons, right? And this is what we kind of meant on the previous slide by talking about sensitization of a response, because now this pattern of activation in some pool of neurons is easy to reproduce now because there has been some change in the strength of the interconnections or synapses between these neurons now, so that activating any one of those neurons is likely going to be sufficient to cause a chain reaction that activates all of those other neurons as well, right? So all of these neurons essentially start working together as something like what we might call a functional whole, right? The idea is that it's no longer just a series of discrete neurons that all kind of influence each other, but they become a collective in a way. And that activating part of that just creates automatic activation that spreads across all of them because of this high degree of sensitization of their responses have occurred because, again, changes in the synapse because of Hebb's law. But there's a different word for it that we're now going to use. We're going to call it potentiation, right? So to say that these neurons have been potentiated, it means that they are now a little bit more prepared or ready to respond to a stimulus. And this change in potentiation will persist for a long period of time. Hence, we get this term, now a very important term in understanding neuroplasticity, called long-term potentiation, or LTP. Right. So let's talk about LTP. Again, we're going to use this as a way of talking about synaptic changes here to explain memory. First point here, specificity. So once we see that, there's going to be, we need to remember that there is this idea of spatial and temporal summation that occurs, right? And the idea is that a given neuron might be receiving input from many different other neurons. So its axons are receiving input from a variety of different, its dendrites are receiving input from a variety of axons, right? And when you start to see changes in these synapses of all of these many axons and dendrites together, it's not about all of them becoming strengthened over time, but only the certain ones that are part of that response, right? So there's a highly specific mapping that is just about the specific axon onto a specific dendrite that can become potentiated in this way. Now, we also have another phenomenon called cooperativity. So this is where that summation effect comes in, especially now spatial summation. So if we have two axons coming together to a dendrite on a postsynaptic neuron, and we repeatedly activate both of those axons onto this postsynaptic neuron, not only are we going to get the spatial summation, and of course, repeated activation is going to give us some LTP because of Hebb's law, but the combined effect of two axons coming together onto the postsynaptic neuron will give a stronger postsynaptic response in terms of the LTP than if it was just a single neuron repeatedly firing over again. So there's something about combining inputs like this in long-term potentiation, this cooperativity, that these two incoming axons are cooperating to even further strengthen these potentiation responses. And then the third one is called associativity, right? So in this case, what's happening here is that if we have these two inputs coming in, but one is a weak signal, right? Remember, we have to talk about graded potentials, right? And so sometimes you have combined graded potentials where a single graded potential might be insufficient to trigger an action potential in the postsynaptic cell. But because of spatial summation, we can have two graded potentials coming in and summing in their combined activity to trigger the postsynaptic cell. But it's often the case that of those two graded potentials that are arriving at the neuron, one is a pretty strong signal and the other one might be a weak signal, right? And so what happens is that because of this cooperativity effect, now it's possible to create such a potentiation here that the strong signal is essentially helping to form LTP for also that weak signal that's coming in, so that later, if it's just the weak input coming in, that weak graded potential could still be now strong enough to create a postsynaptic response, right? And so now this whole system is responding even to very weak inputs. And that's an important part of potentiation, right? Is that now the system becomes somehow very sensitive because even a very weak input will generate a reaction. Whereas previously the weak input by itself is not enough to trigger a postsynaptic response. But now because of the association it forms with a stronger input, now it can actually elicit the response by itself. Here is a couple of diagrams that are showing us exactly these two things here, specificity and associativity. So here we see two pathways coming into a postsynaptic neuron. So pathway number one is a cell with an axon that is synapsing onto our postsynaptic dendrite here. And it's repeatedly active. So we're going to see strengthening of that particular synapse. But meanwhile, path two is inactive entirely. And therefore, it will not show any change in that synaptic strength. But if pathway number two, instead of being totally inactive, is really just sending a very weak graded potential, then if it happens to be paired with this other pathway that is sending a strong stimulus, then not only is the strong stimulus resulting in LTP on the postsynaptic cell, but then that weak stimulus generates LTP that would be more than if it was not paired with a strong stimulus, right? So, and again, in that case, there's this association that occurs between those two pathways, combining their input onto the postsynaptic cell. So we can see here some of the patterns of potentiation that are possible, right? So again, just to back it up there, they're very specific, but neurons can still cooperate or associate their inputs onto postsynaptic neurons to get these effects. But so what's really going on with the synapse here? Well, we're going to look at a couple of different things, some neurotransmitters here, glutamate and GABA, right? And so what's actually happening in the postsynaptic neuron? So let's also remember way back in the chapter on the nature of synapses and receptors, there were ionotropic receptors and there were metabotropic receptors. Here we're going to talk about some of the ionotropic glutamate receptors, and there are two. They're called AMPA or AMPA and NMDA receptors, right? So let's explore what's happening between the AMPA and NMDA receptors here. So here we see there's a simple synapse. Axon releases molecules of a neurotransmitter glutamate. And then we see two different things happening here. So on the AMPA receptor, the AMPA receptor is a very traditional ionotropic sodium channel that we've talked about before. So when the glutamate binds to the receptor on receptor site on the AMPA receptor, it opens up the sodium pathway and lets sodium into the cell. So we're going to start raising the internal voltage of the postsynaptic dendrite here, all right? But look at the MDA receptor, right? So the MDA receptor has this kind of a channel that can be blocked by magnesium, right? And so these magnesium ions can block these things here, and nothing happens there with these receptors, right? But that's just kind of the brief overview. So let's look a little further deeper in here. So we see, for example, that we can find evidence of LTP happening in hippocampal neurons, right? So what's going to happen here is that if we repeatedly excite these AMPA receptors with glutamate, so glutamate's an excitatory neurotransmitter, it's going to activate those AMPA ionotropic pathways. It's going to depolarize these neurons, right? Because you're letting sodium into the dendrite, and that's going to trigger action potentials in the postsynaptic neuron, and you get this depolarization, right? But now what happens is that when you get the depolarization in the postsynaptic neuron, what that has an additional effect, besides triggering an action potential in the postsynaptic neuron's axon, it also kicks those magnesium ions off of the NMDA receptors, right? So now that we have cleared those magnesium ions out of the NMDA receptors, then what happens is that the glutamate that actually still activates those NMDA receptors now opens up the pathway, because now the pathway that was previously blocked by magnesium is now open. And what is happening here is that calcium can now enter the cell. So that's what the NMDA pathway allows to pass through, is calcium. So in addition to sodium coming into the cell that's depolarizing it, that depolarization also triggers the clearing of the NMDA receptor so that calcium can come into the neuron now as well, right? But now what happens? What is calcium doing? So calcium activates a protein inside of the cell, right? And proteins are like these building blocks of that determine kind of the structure of things and what's actually happening in the cell. So what this protein is going to do is it's essentially going to start building more AMPA receptors, right? What happens if we get more AMPA receptors? That means there's more receptors on the dendrite to receive all of that glutamate, so that now the postsynaptic cell essentially has become sensitized, because if it can build more AMPA receptors, it's more likely to receive a depolarizing signal from the axon, right? And it also, these proteins also cause a little bit more dendritic branching. So in addition to having more AMPA receptors, of course, we need more dendrites and more dendritic spines to grow those AMPA receptors on. And so by branching out more dendrites and growing more AMPA receptors across all of these dendrites, clearly the postsynaptic neuron is going to be far easier to stimulate now, because you could just dump it with glutamate, and it's going to have so many receptors that are there to respond to all of that glutamate that it's going to be very easy to trigger an action potential in this postsynaptic cell, right? But it requires, of course, that NMDA receptor gets cleared, and then the input of calcium now triggers this internal series of protein signals to cause these essentially what we'll call morphological changes in the postsynaptic cell, right? It's going to grow. It's going to start growing these dendrites and dendritic spines and AMPA receptors, right? So we can see that here in this picture a little bit. So again, we see the basic idea that the AMPA receptor is stimulated, and we're going to get the influx of sodium triggering the depolarization. That then, as it's labeled there, displaces that magnesium that was blocking the NMDA receptor channels, calcium. You can also get some sodium coming in there too, you can see there, but calcium primarily is going to come in. And then what happens is that we're going to build these extra dendritic spines, and that's what we're going to see over here on the right as we build a second little spine that has an AMPA receptor on it, so that now we get additional places for the dendrite to receive more glutamate and therefore be more easily activated. Right? So that's a change to what we're going to say is the postsynaptic neuron, right? In the postsynaptic neuron, that protein signal helps it build more receptors, right? But what about changes to the presynaptic neuron, right? So what can we do to the presynaptic neuron to help potentiate a synapse, right? Well, what happens here is that as we repeatedly stimulate that postsynaptic dendrite, not only is the dendrite growing, but what happens is that the dendrite releases a neurotransmitter of its own. These are called retrograde neurotransmitters because they travel backwards, right? So normally we're thinking about neurotransmitters going from the axon to the dendrite, but these are retrograde transmitters are going from the dendrite back to the axon, right? So this signal that goes from the dendrite back to the axon causes changes in the presynaptic neuron, right? So what can it do to the presynaptic neuron? One thing it can do is it can change the threshold of that neuron's action potentials, right? So we've previously talked about this idea of graded potentials that have to sum themselves together in order to be sufficiently strong to cross the neural firing threshold, and that then triggers the chain reaction of depolarization traveling down the axon, right? That's way back in the chapter on action potentials. But if we lower that cell's firing threshold, then it's a lot easier for the presynaptic cell to fire action potentials. And of course, if it's easier for that to occur, then of course it's more likely to keep sending signals to the postsynaptic cell. Something else that this retrograde transmitter can do is trigger axons to release more neurotransmitter, right? And so of course, if we're releasing more neurotransmitter, we're going to have a greater likelihood of stimulating the postsynaptic receptors, especially if we're simultaneously building more of those AMPA receptors. So you get, think of these two things happening at the same time, is the dendrite's building more AMPA receptors while a retrograde signal is telling the axon, hey, make more glutamate, right? And so now we have a lot more glutamate and we have a lot more receptors for glutamate, and together that change in both the presynaptic and postsynaptic cell is going to potentiate that synapse, right? And of course, additionally, growth processes. So just like we described, if we're going to start branching off more dendrites and more dendritic spines, that's not going to do much if we don't also do something to the presynaptic axon so that it branches off more axon terminals to connect up to all of those extra dendritic spines. So yeah, we need that, right? So what we've done to the postsynaptic cell is grow more dendrites and more AMPA receptors. What we've done to the presynaptic cell is grow more at the axon branches and more neurotransmitter production, but also even sensitizing that presynaptic neuron to fire more action potentials because of the change in its threshold, right? So it is in this way then that we can really metabolically change the synapse to make it sensitive, right? We also would imagine that, we were talking about habituation with the sea slug, that the opposite can occur, right? That when you have a lack, now with habituation, we're talking about repeated stimulation, but there were those times where we talked about where you're not using a synapse, and that because a synapse doesn't get used, there can be pruning processes. So you can kind of imagine in some ways the opposite of this occurring, right? That because synapses are not getting used, that means that there could be metabolic processes that say maybe we need to get rid of some of these dendritic spines and start shutting down some of these AMPA receptors because they're not there. We don't need to use them for anything. So you see a decrease in the sensitivity of a synapse over time. But all of this is really just about sensitizing the synapse here, right? And that's of course the essence of Hebb's law. Repeatedly activating a synapse sensitizes it or potentiates it through these processes, right? With glutamate and calcium and the protein messengers and the retrograde neurotransmitters. So, all right, I think that was the last slide, right? So, all right, we are done with that chapter.